Welcome to a Bay Area Skeptic Skep Talk.
Good evening, and welcome to Skep Talk, the monthly speaker series hosted by the Bay Area Skeptics. Before tonight's presentation, a few words about Bay Area Skeptics. As North America's oldest skeptical organization, we are dedicated to promoting critical thinking, logic, and rational thought. In the 1970s and 80s, scientists such as Carl Sagan and magicians such as James Randi established the scientific skepticism movement out of concern about the popular fondness for pseudoscience and the lack of public science literacy. Scientific skepticism embraces evidence-based civil discourse on health, education, consumer protection, and especially the investigation of extraordinary claims. Using the scientific method, we hope to promote skeptical inquiry to reduce irrational thinking in society. This talk is supported by Bay Area Skeptics members such as you. Please visit us at baskeptics.org or join us on Facebook, Twitter, and Meetup. Consider supporting us through convenient monthly donations via Patreon. Patrons receive premiums such as discounts and free t-shirts for our annual Skeptical Conference. But more importantly, members help us make the world just a little more reasonable and rational. And now, tonight's speaker.
Well, good evening, everybody. I'm Dave Allman-Smith. Glad to have you with us. Please mark your calendars for our March Skep Talk featuring Daniel Loxton. That will be on Thursday evening, March 11th. We are delighted that Dr. Nathan Lentz is joining us tonight and will be bringing us up to date on some exciting developments in genetics that were not even dreamed of when I did my graduate work in physiology. I could take time to tell you of Dr. Lentz's accomplishments. You know, his professorship at John Jay College, his books, his science publications, his periodical articles, but that would take away time from Dr. Lentz's presentation. Instead, just Google his name. Go to his Wikipedia page. There you will find the impressive details and the address to his webpage. Okay, during Dr. Lentz's presentation, type any questions you have over in comments, and at the end, I'll read selected questions for Dr. Lentz to answer. Dr. Lentz, take it from here.
Okay, well, thank you very much for that non-introduction. I'm joking because actually I find the introduction section of these things the most embarrassing and awkward part of the presentation. So I appreciate that very much. And thanks for the invitation from Bay Area Skeptics to talk about some of my research. This is going to get pretty technical, and so I pride myself on being able to communicate complicated topics to a broad audience, but this is really going to push those skills to their limit today. But I'm going to do my best to try to boil down the most important parts of this work. I pitched several ideas when they invited me to come speak, and all of them on big pictures of human evolution. But my research was the narrowest, most specific talk I could have given, and that's the one they wanted. So if everyone finds this a little too technical and boring, you'll have to blame the hosts, because that's what they wanted me to talk about. Anyway, it is a pleasure to be here and to talk about the work from my laboratory, where we've discovered what we think is a new mechanism of how microRNA genes can be created through the weirdness of genomic rearrangements in the human genome. Okay, so let's get started. The idea, the compelling question that motivated this work is basically human uniqueness. And anybody who follows my work might be a little surprised, because I spent a lot of time writing about how humans are very, not so different than the other animals, as was the title of my first book. But there's no denying that humans really are a unique creature, a unique species. We do a lot of things that no other animals do, from doing calculus and writing poetry to building skyscrapers, and really we transform every environment that we enter into. And we know a lot about human uniqueness in terms of how we're unique, our anatomy. We're not just apes that stand upright. We have a lot of peculiarities in our anatomy. We develop in very peculiar ways. We have a very extended childhood. We essentially never really mature. We're domesticated species in that way. We have very different behavior. Our brains work different, and really our sociality is the root of all of this, is quite different as well. But so we know how humans are different than other creatures, but we know almost nothing about the genetics of this, about how our genes really give rise to this very unique way of being that we have. So I often say that human uniqueness is a near complete mystery when it comes to genetics. And in fact, we do know by studying the regions of the genome that have been the most under the watchful eye of natural selection in our species, really no big bombshell discoveries have been made. I mean, we've done great work, but it's been small tweaks of all really that we've seen, either to genes or to regulatory regions of the genome. Really no like earth-shattering results in studying our genome. So how we got so different from the genetic point of view, it remains a mystery. And just to sort of say this in a simple way, the vast majority of our genes, really almost all of them, have one-to-one orthologs in the other African apes. And an orthologs are genes that have a shared evolutionary history. So they're counterparts. We have almost perfect counterparts of all of our 20,000 protein coding genes. So this is a story of human evolution. That's what we're going to be talking about today. So this is our branch, which is the Homo sapiens lineage, and that branched off from the chimpanzee branch about between 6 and 7 million years ago. So we're going to be talking about the chimpanzee genome. Most people know that chimpanzees are our closest relatives, but what many people don't know is there are actually two species of chimpanzees, the bonobos and the common chimpanzees, and these are equally related to us because they're sister groups that have diverged from each other only about 1 million years ago. Going further back to about 10 million years, we have a common ancestor with gorilla, and these comprise the African apes. And that's really what we're most interested in, is how did humans become so different than the other African apes? There are two species of Asian apes as well, the orangutans, and then gibbons, the lesser apes, the gibbons. And now we're going back more like 18 million years. So these are called the ape clade. These are all the apes that are currently living. There are many extinct apes species, but these are the extant apes. So these are the genomes that we now have sequenced and we have access to. So if we're studying the genes of involved in human uniqueness, we need to do a little primer on what genes are and how they're expressed. Many people, this will be review for you, but DNA is basically a set of instructions for building a protein. So if you have the DNA in the nucleus of a eukaryotic cell, what it really is the A, C, G, T, all of those sequence of nucleotides is really just coded information for building a protein. That's really all it is to it. But it turns out that the recipes for building proteins comprise only 1.5% of our genome. So all the DNA that we have in our cells, the vast majority of it does not code for protein. So what is the rest of it doing? Well, a little bit of it codes for RNA. And RNA is what helps us build, it does the work of building the proteins from those kits. And we're going to talk a lot about RNA today. And then a much bigger fraction of it, about a quarter of our genome, is what we call regulatory sequence. And these are pieces of DNA, large pieces of DNA, that help control when, where, and how much each gene is expressed. And that's really the biggest expansion of genomes over time, is in how they control themselves to be expressed in different cells at different times. And that actually takes a lot more information than the simple recipes of how to build the protein. But if you add up all these numbers, you have 25, 26, maybe 27% of our genome, and that's it. The rest of it, 75% of our genome, really doesn't do anything at any one point in time. It has potential to come to life later, and that's a little bit about what we're gonna learn today. Now, RNA, there are many kinds of RNA, and generally they all have to do with gene expression. So the most famous RNA is the messenger RNA, which takes the instructions from the DNA out to the cytoplasm. If we're talking about a eukaryotic cell, here it is here, the mRNA molecule. It takes the instructions from the DNA out to build the protein. And this is essentially a perfect copy. So we call this process transcription, where you make RNA from DNA. It's just a perfect copy of what you have in the DNA, ideally speaking. And then when the RNA reaches the ribosomes, which are the factory for making the protein, that code is translated from an RNA code or a DNA code into the protein code, which is, of course, amino acids. So mRNA is the most famous, but the transfer RNA, that's this molecule right here, is what brings those amino acids to the ribosome. Here's the ribosome here that is sliding along the mRNA and building the protein. So this is an RNA molecule, tRNA. These ribosomal subunits are made of RNA as well, and of course this is messenger RNA. So those three are the big three, but there's actually many other kinds of RNA. They all have something to do with helping us use our genes in very precise ways, getting them expressed at just the right amount at just the right time. Well, if you look in the literature about what we've learned about how our DNA has changed over the last few million years to give rise to humans and our uniqueness, what you find are large studies, and this is the Kagan study, which I'll be talking about later today, that attempts to find all the regions of our genome that have been under natural selection, that have evolved the most rapidly. And this is a groundbreaking study, and we refer to it almost daily in my lab. And it identified the 200 genes that were most positively selected, the 200 most stably selected. But there's really no surprises in there. There's no genes that we don't also have with the other apes, that we don't share with the other apes. Slight tweaks is really all we're talking about. We have another data set that we talk about a lot in our lab, and these are the genes that are involved in domestication. One theory of human evolution is that we're the product of domestication, that we are we've domesticated dogs and cats and livestock animals and even plants and things. We also think we've domesticated ourselves, and the genes involved in that are somewhat known. But again, no big surprises. These are genes that we share with a lot of other creatures that just have slight tweaks in the human versions. So my lab decided to take a new approach to this question of human uniqueness. Is why don't we try to study the genes that we have that are unique to us, so that are really just human-specific genes. And this introduces a topic I have to now tell you about called orphan genes. This is not really a technical term. We don't use this term too much anymore, but it's the older term that most people are familiar with. Well, orphan genes represent one of the biggest surprises of the genomics era. So as you know, 20 years ago, last month or the month before, the first draft of the human genome was announced. So I was already in graduate school at this point when we had our first full draft of the human genome. Since then, we've now sequenced millions of individual humans' genomes and hundreds of thousands of other species. We are in the genomics era. We have a lot of genomic data. We're swimming in genomic data. But one of the biggest surprises, and this actually goes back to when the yeast genome, the first eukaryotic genome that was sequenced, when the yeast genome was sequenced, the biggest surprise was, at the time, this was in the 90s, 1/3 of the genes that were discovered had no relationship with any other genes that were known in any other creatures. 1/3 of their genome, 1/3 of their genes seem to have come from nowhere with no evolutionary past. And that was a very big shock, because everyone always assumed that all genes evolved from each other, just as all organisms are descendant backwards in time towards a common ancestor of all life. They thought genes, you could just map them all back that way as well. And that turned out to be not the case. A lot of genes in yeast were discovered as if they came out of nowhere. Now, part of the problem was we didn't have a lot of genomes sequenced yet. So as more genome sequences became available and more comparisons were made, a lot of those genes were de-orphaned, and we were able to figure out where they came from. But it's still true that about 20% of the yeast genome were actually are comprised of orphan genes that don't have any relationship to genes in other organisms. We don't use the term orphan gene anymore. We say taxonomically restricted genes. And this field has exploded over the last 20 years. Now, taxonomy is the organization of organisms into groups. And so you can have genes that are specific to a species, for example, human-specific genes, or gibbon-specific genes, or whatever you want to say. But you can also have genes that are specific to a group of organisms. You can say the chimp-human clade. So this is a group of organisms that share some genes that others don't have. Or maybe there are ape genes, or there are primate-specific genes. There are even mammal-specific genes, and so on. So you can have these genes that are restricted at any level of that hierarchy, including the species level. And this can be a large fraction of the genome. So for example, in Drosophila are fruit flies, and they are the most commonly studied organism when it comes to orphan genes. After sequencing many species of Drosophila, the most famous one is Drosophila melanogaster, but there's many species of Drosophila. About 18% of their genes are only found in that genus. They're only found in Drosophila, not in other two-winged insects or anything else. We found at least or approximately 60 human-specific protein coding genes. So the humans have at least 60 proteins that are not found in any other organism that we know of. And that's dated information, so it's probably larger now. The take-home point here is that the big surprise of this genomics era is that we have a lot more uniqueness in every genome than we thought. And there's a lot of genes that do not have a very deep evolutionary past. Now, some of those genes are called young genes, and that's different than another class of genes, which I'm going to cover on the next slide. But young genes are those that have just emerged recently, and there's a couple of processes that give rise to these. Some genes simply duplicate, and then they diverge so rapidly that they're almost unrecognizable. Some genes fuse together. Two genes can fuse and form one. Genes can split, and so that's called fission. So you have fusion-fission events, and these generate chimeras or things like this. Horizontal gene transfer is a fascinating way that genes can enter an organism where from two unrelated species, a gene can actually move from one species to another, usually by way of a virus. And my favorite example of this is actually in ticks. So ticks have some genes for fighting bacteria. They have basically antibiotic genes that they have in their genome. Well, we've actually found that they got those genes from the bacteria themselves via a virus infection. So they acquired new genes from bacteria, and then they use those genes against the same bacteria. So that's a pretty dramatic example. Usually they're more subtle. This is not a common way that we get new genes in humans or any animals really, but it does happen a lot in the microbial world. And there's other mechanisms as well. But I'm not even going to talk about these kind of young genes. There's an even more dramatic way that you can get new genes in the human genome, and it's called de novo gene creation. This really is the kind of thing that we would have thought was laughable 20 years ago. Even 15 years ago, when it was first being discovered, it was thought to be just a really a fluke thing, of it really not a common occurrence, but we now know that it happens a lot. So the technical definition of a de novo gene is a protein gene that was made from previously non-coding sequence. It really is a gene from nothing or a gene from junk. And there's several types of these, and I don't want to go into these. I wanted to put it up on the slide for those who want to read more, but that gets pretty technical. But let me just say that the reason we now know that this is possible is that the genome, all of that junk that I told you about before, the 75% of our genome that really doesn't code for anything, it turns out that it is spuriously transcribed into RNA. And this is what we found over the last really, again, about 20 years, is that transcription is actually rather sloppy. There's a high rate of just sort of background noise. Where random sections of the genome are being transcribed into RNA. It doesn't really do anything, but it's sort of there in the background. And because it's there in the background, evolution has it as within its toolkit. The most common spurious transcript is just simply called long non-coding RNA. It doesn't seem to code for anything. It's just there. However, what we found is that ribosomes, which are the factories for protein synthesis, are often associated with these. And we've even detected what we call spurious translation. So random peptides, random proteins are actually being made in every cell of your body to no end, to no purpose. But they do have a purpose over evolutionary time, is they add to that toolkit of potential new genes or new proteins. And the way that this works is, it turns out that genomic rearrangements are actually very, very common. And I'm going to show you an example of how common this is on a human chromosome. And that leads to what we call tinkering, where the DNA is just sort of rearranged and duplicated and moved around and shuffled around. And what this does is create transcripts that are different, and some of which may sort of land on a functional product just by pure chance. And there's two things you need to get sort of a gene from nothing or a gene from junk. You need an open reading frame, which is a sensible coded instruction for a protein, which is something the cell can actually read as instructions for a protein. It doesn't have to be a functional protein yet, but just something with a beginning and an end and a middle. And then you also need the sequences to initiate that spurious transcription and then hopefully ramp it up further if you want to really do something, because generally new genes tend to have very low expression. Okay, so that's what you need. But if you're doing this and you're detecting all of these spurious transcription, spurious translation, how do you know it means anything? How do you know it's not just in the background noise? So there's a few tests that we do to tell that. Number one, you have to say, is it transcribed? Is it translated? And does it make meaningful interactions? Meaning, does this protein actually bind to some other proteins and change them in some way? Or does it bind to DNA, for example, and do something there? So does it have functional interactions? And then the most important test of all is it being maintained by natural selection? So is its mutation rate what we would expect it to be for a part of the genome that evolution cares about, that does something useful for the organism? This is just a graphic of this. I don't have to spend too much time in this, but the point is that there's various ways where you can have sort of background transcription of DNA into RNA, and these ribosomes can actually jump on to the RNA product and make random peptides. And these peptides might actually do something. It would be weak and they would be promiscuous. And the word promiscuous in this context means not very specific. It can do all kinds of little things, make interactions and do random things. But once it starts doing something, no matter how weak or how promiscuous, that something might have an effect on the organism. And once it has an effect on the organism, on the cell, it can be selected for or against by natural selection. And then when that happens, when evolution starts watching this gene, then it can start to take shape. And it would get more refined and more refined and more refined as the bearer of this new gene gets the benefit of its function. So the first surprise is that we have these things. The second surprise is that they do stuff. And then the third surprise is that these de novo genes, which really come out of nothing, can become important to the organism very, very quickly. And as I said, most of this work is done in Drosophila, but just a couple of recent studies have shown in a fairly quick amount of time, these genes can take on very important functions for the organism, even essential functions. And in insects, what we've seen, the most common way that new genes become important is in their development, meaning their metamorphosis. So these fruit flies undergo a pupal stage with, and they come through as winged adults. That's generally where the new genes tend to be most active. And the parallel to that in us, in mammals, is embryonic development. And we see the same thing, that new genes tend to most often have an effect in embryonic development. So just keep that in mind for later. A couple of human examples, and then I'll move on. There's a gene with a very unfortunate name because it was named by an annotation project rather than by a lab that discovered it. But this gene, it's called FLJ33706. It's highly expressed in the brain, and it's linked to nicotine addiction and Alzheimer's disease. I mention that simply because that means it's doing something in the brain. It has no similarity to any protein in any other species. We know a little bit about how it was created. I don't want to go through all the technical details here, but it has 6 exons. The first one is not expressed, but these were created through a bunch of random insertions of this transposable element, which is a mobile piece of DNA that is known very well for jumping around the genome. We have more than 1 million copies of this guy jumping around through our genome over time. And then a bunch of point mutations created the open reading frame. And here's the open reading frame here. So this is the start codon that tells the cell when to start building a proton, and here's the stop codon. So these point mutations created a start codon, and they disrupted early. So we basically have mapped out how this gene came to life from lifelessness. And it's an interesting story. Most importantly of all, though, is it shows signals of selection, meaning natural selection cares about this gene. It must do something important for us, not just giving us Alzheimer's disease and getting us addicted to cigarettes. It must be doing something in our brain. We don't know what yet because it's very hard to study, which is actually brings me to an important point. A lot of these orphan genes are very hard to study. The reason why is our typical way of studying genes is to look at what they do in organisms that we can manipulate. I know we can take the gene out and see what happens. You can take the gene out of a mouse or a fruit fly and watch what happens. Well, there's a problem with doing that here because this is a human-specific gene. So by definition, you can't use any other organism to get at your answer. And they're going into a human brain and dissecting out the gene from a living human. Most people would consider that a violation of ethical principles. So we actually have limited toolkit in how to study what these genes do, but we do know that they do something important. Okay. Another example. So there's a gene that stands for an embryonic stem cell related gene. So this is a gene that's involved in keeping stem cells pluripotent, which means the potential to become anything, any kind of cell in the body. So this gene is very important for that. Remember, I said embryonic development seems to be a thing that they're commonly involved in. We know a little bit about how this one came from a endogenous retrovirus. So these are little retroviral genomes that live in our chromosomes, pop out every now and then. And wreak havoc. In this case, the havoc that they wrought created a new gene, which turned out to be important. And there's other ones too. This is actually a cancer-specific gene. So this is a gene that's involved in chronic lymphocytic leukemia. So that's a blood cancer. Nobody really knows exactly how this works, and we don't even know if it's the protein or the RNA that does it, but it does seem to drive CLL, which makes it a good, what we call druggable target. Pardon me. Okay, anyway, that's some of the work in the field up till now. So now that brings us to what we've done. So you came here to hear about what my lab has been up to. So we decided to take a different approach because most of the work that's been done on this starts with either proteins or at least messenger RNA and tries to work backwards. But we wanted to take an unbiased approach looking only at DNA. So what we did was we wanted to look at unique DNA elements in our genome that only humans have that our closest relatives don't have, because we didn't care if it would code for a protein or code for an RNA, or maybe it was just another kind of functional genetic element of DNA. So we wanted to take that unbiased approach. So we used a program called Mummer 4.0 to align human and chimpanzee chromosomes and just look for differences. It sounds really simple. This program was actually not designed to do this. It was designed when you're building drafts of the same genome and you get more information and more drafts as you line them up to build the composite map of a chromosome. So we're using it for a slightly different purpose. We're not the first to do this. We didn't invent that idea, but it's picking up steam, I think, and I think you'll see why. So this program is useful for identifying regions of what we call non-alignment. So the chromosomes are not lining up. There's things missing. That's at least the potential for something unique in one chromosome or the other. So we asked the program to find these regions, and then we programmed it to say what's nearby, what genes are nearby, and give us these lists of genes that are in these unique areas. And then what we did was we looked for orthologs. I think I said what an ortholog is a gene that exists as counterparts in other organisms with a common evolutionary past. And we did that with just BLAST searching, which is sequence mapping, or sequence searching, excuse me, or matching. And so we're looking for these unique elements on chromosome number 21. Why did we start with that chromosome? Because it's the smallest one. So we decided that we would work our way from the smallest chromosome all the way up to the largest chromosome, which is chromosome number 1. Long story short, we're still on chromosome number 21. We haven't even made it to the next one yet. And I think you'll see why. And just for anybody who's interested in technical details, we were working with HG38, which is the most recent draft of the human genome. And comparing it to the chimpanzee genome, again, the most recent draft is PanTro6, which stands for Pan-troglodytes, the scientific name of the chimpanzee. Okay, so here's what we did. This is basically the first result that we had. So we line up the chromosomes. So this is the coordinates for human chromosome 21 going from 0 to 45. And these are megabases. So those are millions of base pairs. That's what the unit is there. So base pairs are the nucleotides that make up the DNA, the A, G, C, T, C, Z, G, T, and so on. These are the megabases there. So you'll notice that what the program does, what Mummer does is it attempts, so it looks for a sequence. It says, okay, here's a sequence we're looking for, and it finds it, and then it attempts to extend that sequence in both directions as far as it can while still matching your criteria. And in our case, the criteria was similarity, a minimum amount of similarity. So, and then it matches all those up and lines them up. So when we lined up human chromosome 21 with chimpanzee chromosome 21, you'll see from about this point onward, sorry, I should be up here, has very, very good alignment. So most of what we were looking for in the chimpanzee genome, most of what we're looking for from the human genome, we found it in the chimpanzee genome just fine. However, at this point, sort of in the middle here, there was almost nothing matching. Well, it turns out that this is the centromere. This is the central region of the chromosome that is structural. It's not a coding region. And so that is uninteresting to us. It's sort of the middle point of the chromosome. But what we noticed on the other side, it also did not match up very well at all. So there's all of this human DNA in this area where we can't find anything corresponding to it in the chimpanzee chromosome. So that was weird. That was a real head scratcher, because I did not expect to. I expect to have tiny little breaks. That's what we were looking for. But then I thought, well, DNA does move around from chromosome to chromosome. So instead of only looking in chromosome 21, we should be looking in the whole genome of chimpanzees. So we're going to stick with human chromosome 21, but now we're going to ask it to find the DNA anywhere it can in the chimpanzee genome. This clarified things a little bit. So it firmed up what we call the long arm of chromosome 21 on this side of the centromere. Here's our gap again for the centromere. And you see all these fuzzy lines here that represent other probably duplicated regions of the same DNA on other chromosomes. It doesn't really cloud our analysis. But the fascinating thing is the short arm of chromosome 21, so here's the coordinates here, is still not very well aligned. So this tells us that there are tons of regions of human DNA over here that there's no equivalent of in the chimpanzee genome, at least no equivalent that meets our criteria of length and similarity. And you can set these criteria and parameters, and we spent weeks, probably months, tweaking those to making sure we weren't missing something. Okay, so there's all this DNA on the human chromosome 21 that we can't find on the chimpanzee. But that doesn't mean that it's unique to humans. It could be that chimpanzees lost it, right? So you could have either problem. Either humans have this extra stuff or the chimpanzees lost it. So what we decided to do was basically just do the same thing again, but do it with the other apes. So what I did was we mapped human chromosome 21 to the entire genome of gorillas, chimpanzees again, and bonobos, which are the other species of chimpanzees. And here's the centromere, here's the long arm of chromosome 21, and you see it matches up really, really, really well. So even with the human genome, you see it's further from gorilla, but remember that we're separated from gorillas by about 10 million years of evolution. So they're more distantly related to us. So we expect a little bit more gibberish here. But once again, the short arm of human chromosome 21, we simply can't find equivalents for it in any of our most closest relatives. But yet, if we look at those close relatives to themselves, it's fine. So this is now lining up chimpanzee chromosome 21 with gorilla chromosome 21. And you notice I didn't even feed it the whole genome. I only gave it chromosome 21, and you'll see that the chimps and the gorillas have it's not an identical, but really through the entire length of the chromosome, it matches up really, really well. So we are looking at a human difference here, because remember that they're separated by 10 million years of evolution as well. So these two are more distantly related than we are to chimpanzees, for example, and yet you still get much better alignment. And the same thing is true if you go all the way out to orangutan. So this is chimpanzees with orangutan. So now you're at 12 to 14 million years of distinct evolution, and you get really nice alignment through the entire length of chromosome 21. But yet with humans, if I back up a little bit, you'll see this huge gap doing the same kind of alignment. So really the short arm of human chromosome 21, something weird is going on there. Okay, so now let's zoom in a little bit. We're going to keep comparing to chimpanzees, but now if you'll notice the coordinates, we're not doing the entire chromosome, because we sort of know the picture that the long arm is fine, but the short arm. So now we're looking basically only at the short arm. This is the centromere region, and this is the beginning of the long arm. Now we're going to zoom in on our analysis, and we tweak the parameters a little bit. So we're seeing some more of these inverted regions, the blue regions. And what we notice is that there were some microRNAs in this region. Now, there were lots and lots and lots of gaps here. And so you might be asking, why are you concentrating on right here? Well, most of these gaps, this whole region, for example, has almost no genes in it. It's a gene desert. And you have chunks of this on all of our chromosomes where there's just really no genetic information. It's just junk DNA with a few islands of information in there, but it's mostly nothing. But in this region here, we did have genes, and we did have genes that we found to be orphans, meaning these microRNA genes, which I'm about to tell you a little bit more about, they were mapped, they're annotated on the human genome, and we know that they're expressed and they're functional. I'll talk about that later, but they have no equivalent in the chimpanzee genome. It's never been mapped in the chimpanzee genome. So we found families of orphan genes right in this region. I'm going to zoom in even further now to just this region. Again, we're comparing human chromosome 21 with the chimpanzee genome, entire genome. So you see these huge sections of just no alignment, of just really human unique DNA. And what we found is there are 3 families of microRNAs. Now, I'll tell you what microRNAs are in a moment, but to make a family of microRNA means you have to have identical products. So MIR6724 has 4 genes that all encode the exact same thing. So it's basically 4 copies, almost identical copies of the same gene. And there's 2 copies of MIR3648, one here and one here, and 2 copies of MIR10396. Now, just to let you know, so basically we have 8 microRNA genes in this region, none of which are found in chimpanzees or any other ape, any other organism at all. I'm going to cut to the chase and tell you right now that this one is not functional. So this gene family, these 2 genes do not encode anything that we believe is functional. We have no evidence that it expresses anything, so we don't think it does. But these 2 definitely do. They've been found in screens of all kinds. So now let's take a look at this region using what's called the genome browser. I mean, any of you who do genetic research have used the genome browser. So here's a schematic of chromosome 21. Here's the long arm, and we're focused right in on this region called 21p11. And I put arrows here on the genes of interest for us. So you'll notice this region has a bunch of genes in it, microRNA genes, as well as other RNA genes, which I'll be talking about. There are no protein coding genes in here. So right off the bat, I was really patting myself on the back for not taking a protein biased approach, but just looking for novel genetic elements, because we would not have found this if we were looking for proteins or messenger RNA. We were taking an unbiased DNA approach, and we found all these orphan genes. Down here, actually, I'll talk about these on a future slide. So these are called CPG islands, and they're evidence that these are active areas of the genome. I'll talk about that later. But first, we just have to talk about what the hell microRNAs are. I've talked about discovering these genes, but I haven't told you what they are. So let's do that now. Okay, what are microRNAs? Well, microRNAs are one of the most recently discovered biomolecules out there. So in fact, when I was in college in the 1990s, this was not a thing. It was not in textbooks. It had been discovered unbeknownst to most of us, but it was thought of as a curiosity in nematodes or something. But it was in the early 2000s when this was recognized to be important microRNA genes. So what they do is they regulate the expression of other genes. So they're part of the gene expression apparatus. And the way they do that is they target messenger RNA, which remember codes for proteins, and it targets them for destruction. So it's a way to turn gene expression down of its targets. And a single microRNA can turn down expression of dozens or even hundreds of genes. So they're very powerful gene regulators. And about 60% of all of our protein coding genes are subject to this kind of regulation. So microRNAs are very important in regulating expression of other genes. These are only found in plants and animals. So they're recently discovered, but they're also a recent evolutionary innovation in plants and animals. And they work so differently in plants and animals that we actually believe that they evolved separately, that they're independent convergent evolution there, because they just have too many mechanistic differences to have been evolved once and then diverged. This is what a microRNA looks like. It's a very small molecule. It's very simple, compact structure, and we call it a hairpin because it has this double-stranded nature to it, and then a loop. And this loop is single-stranded RNA, and then you have this double-stranded RNA here. And this is the targeting region of the molecule, so where it finds its targets, and specifically this seed region of 7 or 8 nucleotides long corresponds to a structure in a messenger RNA by just simple sequence matching, sequence complementarity. It finds its target and then destroys it. So if you have a gene for a microRNA, it expresses this larger microRNA, a larger structure, which then gets chopped down to the hairpin, and then that hairpin gets further processed into its targeting version. But there's another way you could have a microRNA gene, and that's within the intron of a protein coding gene. So these are actually genes within genes, but they still can be processed the same way. So this lariat structure gets spliced out of a messenger RNA, and then it gets processed into a microRNA. So it doesn't matter which way. All the genes we're looking at today are of this variety. The point is that once you get this targeting apparatus, this can then go and do its work, which is to chop up other messenger RNA. So here's what a microRNA would look like, and here's its target. This is a messenger RNA. And you see it's found it because of sequence matching here, and it's going to now destroy this messenger RNA. It's going to prevent translation. So this is the ribosome trying to build a protein out of this, and this one's sitting in the way. And then it'll also begin to chop up the messenger RNA through a process called de-adenylation, where it chews up that messenger RNA from the outside in. Okay, so what we found is these microRNA duplicated genes are all here, but as I said, they're duplicated. There's 4 copies of MIR6724, and MIR just stands for microRNA, and there's 2 copies of 3648 and so on. So, but everything wasn't just perfect, 2 copies here, because this cluster is not exactly the same as this cluster. Is this 3 clusters here, or here's another lone one? And then these 2 are way over here. So we knew we were looking at something more complicated than just one thing copied into 2, that there was something else going on. So we decided to take a new approach where we looked at this chromosome compared to itself. So what this means is we're looking at, so it's similar to MUMMR, it's actually using the same program of MUMMR, where we take the chromosome 21 and look at bits of sequence and say, find where else that sequence exists on the same chromosome. So it's essentially what you're trying to do is line the 2 chromosomes up to themselves, but in a way that reveals sequences that are duplicated and exist more than once. So here's the centromere of chromosome 21. And once again, the long arm is all nice and tidy and everything's neat and clean. There's really just 1 copy of everything. But in the short arm of chromosome 21, it's just a mess of duplications. And everything you see here that's sort of off the line represents a duplicated sequence, a sequence that's there on the chromosome more than once. So we're going to zoom in just here on the short arm. So if you look at the coordinates, we're going from 0 to 50. That's the entire chromosome. But now we're going to look just at these handful of megabases on the short arm. And you can see what a mess this is. So if you, and since this line represents the chromosome versus itself, it's a mirror image on either side. So it doesn't matter if you look at this side or this side, they should be mirror images of each other. And to get an idea of how many times these have been duplicated, if you just draw an imaginary line horizontally, however many times you hit something at that same point, that's a duplicate because you're at the same coordinate here. So right at this coordinate, there's a duplicate, 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 duplicate. That's how many times that sequence has been duplicated. And you can do the same thing with vertical lines. And you'll notice that most of these are purple, but there are some blues here. And blue represents the other strand. So it's a forward strand of DNA. There's also the reverse. So these are inversions. The same sequence is there, but it's been inverted. But they are much more commonly not inverted. So, and that's going to be important for understanding the mechanism, and I'll come back to that later. Now, if this is the short arm here, this is the really busy section when it comes to duplication. But if you remember, I said before, that is a gene desert. There's not much going on there. So this is where our area of interest is. So I'm going to zoom in yet again on this region. So you can see this formation. When we first saw this, we called it the spaceships, because it looks like spaceships flying in formation. And you can see multiple duplications. You have duplications of duplications going on in this region. So something crazy is going on in this region, and our microRNAs are there in the mix. And this helps us understand why you have 2 of some things, 4 of other things. Regions have been duplicated over and over, and within, so a duplication within the duplication. Now, what this is called is an intrachromosomal segmental duplication. It's a lot of words to just say that it's been duplicated in tandem on the same region. And none of these are inverted, so it does not appear, so that helps us understand what the mechanism is. Because they're not inverted, we know that there's 3 ways that you can get this. You can have transposable elements. So I already mentioned ALU, but there's others. These are jumping parts of DNA that jump around the genome. These tend to be very small. 300 or so base pairs or less. So that's probably not the major factor here because our duplications are in the thousands of base pairs. But there's also recombination with unequal crossing over. You may have remembered from your high school biology class where in meiosis, where you have meiosis is the production of sperm and egg cells, where you have the maternal and paternal chromosomes line up and then exchange material. Well, when that happens, sometimes the crossing over that happens, some DNA is pulled more on one side than the other. And so you end up with extra DNA on one chromosome and less on the other one. So that can lead to some of these duplications. Also deletions, but that's just not what we see here. There's also something called trinucleotide expansion or trinucleotide repeat expansion, which I'll talk about later. So we have multiple possible mechanisms here, and we're going to start to explore them. The one thing we wanted to get a handle on is exactly what the size of these duplications are. This gives us some clue, but there's actually a better way to do this called synteny mapping, where once again, we're mapping the chromosome to itself, and any repeated region is shown on this axis as well as this axis with a line connecting them, showing you how wide it is. So this represents, and of course it's there twice because we have the chromosome to itself, this really fat duplication here represents a very large duplication in tens of thousands of base pairs large. But these small lines represent very small duplications and at great distance. But more interestingly, you can see duplications inside of duplications that must have happened over evolutionary time. And here's our genes of interest right here. So these microRNAs that we found duplicated are all right here in this region, and they've been duplicated through this intrachromosome segmental duplication. And then also over here, this actually could be a transposable element based on the size. The point here is that there's lots of stuff going on in this region that have given rise to these microRNAs. And in fact, actually, when we measure trinucleotide repeats, so these are the combinations of triplets that actually are prone to this expansion, to this duplication process. You'll see our region, this is exactly right, the region we're interested in is enriched for all of them, but some more than others. So we actually think trinucleotide repeat expansion is at work as well. Okay, so let's see, because just duplicating regions of DNA, how does that get you a microRNA? How do you go from just duplicating random pieces of DNA to creating this microRNA? I haven't given you any clues about that. And it's because I have been avoiding telling you about these genes in here. It's not just microRNAs that we see in this region. The MIR, that stands for microRNA. But there are other kinds of RNA in here too. And these other kinds of RNA genes are called ribosomal RNAs. And these ribosomal RNAs have a very particular structure. They've gotten duplicated along with the rest of them, but they also, if you notice, this microRNA maps within this ribosomal RNA gene. And many of them do. Most of the microRNAs that we see actually look like they map right inside of the gene for a ribosomal RNA gene. So let's take a look. So this is what a ribosomal RNA gene looks like. This is specifically the 5S ribosomal RNA. And you'll notice it has a lot of double-stranded nature to it, a lot of loops and that kind of thing. It's a much larger molecule than the microRNA. But you can imagine, I think you can probably see where I'm going here. It doesn't take a molecular biologist to see that it wouldn't be very hard to get a microRNA out of one of these ribosomal RNAs by simply chopping it off, and the slightest tweaks and tugs would get this here. So if you imagine you're copying a chunk of this into a new location, it'd be very easy to generate a hairpin. So that's our working model right now for how these genes got duplicated, or how these genes got created, was through duplications within this array of ribosomal RNA genes. And that's the key part here, is there's not just one ribosomal RNA gene. There's a whole array of them, if you see that. So these were already duplicated long ago, because these arrays are shared with other organisms, but they got duplications within duplications within duplications, and our model is that it created these microRNA genes. Now, we had a little bit of work to do to show that these are really, truly human-specific microRNAs. How do we know they just haven't been discovered? So we took the sequence of these microRNA genes, and we found the closest match in the other apes. So this is the human sequence, HG38. And if you look at the sequence of this microRNA gene and compare it to the other apes, you'll notice a tremendous amount of difference. So this sequence has diverged rapidly from the other apes. You expect 98.8% conservation between the human and chimpanzee sequence, but it's nowhere near that. There's been a lot of evolution of this sequence, excuse me. And if you look at the other microRNA, 6724, same thing. It's not as divergent, so this might have happened more recently or just simply hasn't evolved as much, but there are a number of differences throughout the length of this microRNA. So one way to say this is we don't think these are microRNAs in the other apes at all, but if they are, they don't operate the same way. They are too divergent. They wouldn't have the same targets. They would be a totally different molecule. So either way you slice it, it is a human-specific microRNA that we're looking at. And just in case I haven't said it yet, these microRNAs are expressed because they've been found in many screens by other groups. Also, just to sort of please the reviewers kind of thing is if we were to model what these molecules look like when they take shape. So these are structural predictions, and this is what one of the microRNAs look like, and here's what the other one is. The human version is on the far left, and then the chimpanzee version is next. And you'll see that this doesn't even really take the shape of a microRNA, nor does the gorilla version. The orangutan does look more like a microRNA, but you'll see it has much lower stability. So this is a measure of the thermodynamic stability of the molecule, whereas red is the most stable. And you can see this is not near as stable a molecule. And the same goes for this mirror. So if this is a microRNA, it's not a very stable one. And as I said, it would have different targets. We also are living in a weird time in the sense that we actually have genomes from extinct species as well. So one question I wanted to ask was, do we share this with Neanderthals? So we have 3 Neanderthal genomes that we have access to now. So we got the Neanderthal sequences as well as the Denisovans, which is another extinct hominin. They're sometimes referred to as the Asian or the Eastern Neanderthals. We don't have any full skeletons from these guys, but we do have their genome. And what we notice is that the microRNAs are almost perfectly conserved between all of these. So this predates the split with Neanderthals and Denisovans. Interestingly, 6724 has at least one difference in the human version, but this difference is not in the seed region, so it probably doesn't change targeting, and it certainly doesn't change the folding. So these look like they're perfectly functional, and they're retained in extinct Neanderthals as well. Okay, so the last question is, what do these do, and are these important? Because these are, again, just curiosities. Even if they're expressed, they're nothing more than curiosities if we don't connect them to some kind of function. So what we did to answer that question, we used a program called TargetScan, which predicts the targets of a microRNA. You feed in a microRNA sequence, and it tells you what genes would be targeted. So TargetScan predicted 473 target genes for MIR3648. That's one of the microRNAs that we discovered. And what we were, and if you look at the list of genes, it's one trick you can use to figure out what they have in common is a strategy called gene ontology, which all the genes in the human genome, we have figured out what their functions are, the ones that we have figured out, and we've put them into categories. And what this program can do, or this strategy can do, will tell you, are there functions that are enriched in your list? So these 473, are there some functions that seem to be enriched in that group versus the whole genome? And we found out a lot of enrichment. And the things that are enriched, most interestingly, are in cell-cell adhesion, embryonic development, and neuronal processing. So if you look, these are hierarchical categories, so it's categories within categories, so that's why it looks so redundant. But embryonic development, cell differentiation, and neuron functioning, and basically, these are all of the kinds of things that we would expect to see in a molecule that's important for embryonic development and potentially for evolution tweaking. I mean, this couldn't have turned out better because there's, not billions, but hundreds and hundreds of different possible functions we could have found. And these are very, very specific to embryonic development and specifically development of the nervous system. So, and we also looked within those targets for overlap within the Kagan data set. That's the data set of the genes that are the most selected during human evolution. And there was only 2 genes that overlapped. One of them has to do with cancer, and not much else is known about it. It's a metastasis protein. But the other one is called NSMF, which is the NMDA receptor, synaptonuclear signaling, et cetera, et cetera. But it's involved in our nervous system taking shape during embryonic development. So we thought that was pretty interesting. If we're looking for genes that explain human uniqueness and how we became so different, these are the kind of genes you're hoping to see. If we look at the other MIR, so this is MIR6724, TargetScan predicted 3,000 target genes for this one. That's a little high, so we're not totally sure that those are all real. I mean, we're quite confident that most of them are not. So we only looked at the top 2,000. Even that's probably too many. But if you look at the categories that are overrepresented in those, once again, you see embryonic development, of organs and blood, vasculature and blood vessels. You also see neuronal functioning. Synapses are how two neurons talk to each other, and the genes involved for that are enriched in these targets. And again, embryonic development, neurons, neurodevelopment, cell differentiation. So these are two entirely different microRNAs, and yet they coalesce on the same kinds of functions, at least according to what their targets are or are predicted to be. Of course, this requires a lot of experimental validation. This is all computational work at this point. We haven't been able to get back in the lab recently, but this is very exciting results to us. And if you look at the overlap again with the Kagan data set, so the most strongly selected genes, you'll see again a handful of genes that are from the Kagan data set that are also targets of 6724, and every single one of them have to do with neuronal functioning, functioning of neurons. And you'll see that NSMF was once again on this list. So NSMF connects both of these microRNAs and the Kagan data set as an important gene for human evolution. So we found this to be rather satisfying in the sense that we sort of found what we were looking for, some human-specific genes that may have impacted human evolution. So just to summarize what our results are, the MIR6724 and MIR3648 microRNAs are encoded by two human-unique gene families. These genes were formed through repeated rounds of segmental duplication within an array of ribosomal RNA genes at 21p11. So we have what we think is the mechanism of how these microRNA genes got formed through random segmental duplications. These duplications likely occurred either through meiotic recombination or through trinucleotide repeat expansion. Now, I think it's probably a combination of both of those with some transposable elements sprinkled in for fun, but some kind of mechanism led to these duplications. And these two microRNAs have gene targets that are enriched in gene ontology categories for embryonic development and nervous system function. So we sort of have a complete story here. We're in the process of trying. None of this work is published, by the way, but hopefully it's not unpublishable. And we're in the process now. This is actually the first public presentation of these data, and I'll start presenting it at conferences next month or the month after next, and then hopefully get a manuscript submitted soon. But more importantly, I think, than just this particular discovery, we also, I think, made a proof of concept that genome alignment and synteny mapping, those are the main strategies we used here. They are an effective way to discover unique elements in a genome. And we did this with the smallest human chromosome, but many chromosomes are much, much larger and much more gene dense. Chromosome 21 is not a particularly gene-rich chromosome. There are many other genes, many more genes on other chromosomes. So I think taking this approach is an effective way to find genes that are important in a particular lineage. In this case, the human lineage, which is the one that we're all, I think, most interested in. So that's all I have for you today. I tried to cut down the technical details as much as I could, but I would, at this point, really enjoy taking any questions that you have for me.
Okay, thank you, Dr. Lentz. I have to, in a moment, after the smoke finishes coming out of my ears, it was fascinating. I learned a bunch. I think I will watch this presentation over again on our YouTube channel to get it in its fullest. Are you ready for the first question?
Please.
Okay, let's see. How much bad transcription is there? I'm starting to wonder how this genetics thing ever worked.
That's a great question. So the point is, there's more pointless transcription than transcription of functional regions. So there's a project called the ENCODE project, which has gotten a lot of press. That its goal was to find everything in the genome that was transcribed. That was one of its goals. Everything in the genome that was transcribed and functional. And I don't think anybody expected. I was a postdoc at NYU actually, and our lab was considering joining as part of the project. We didn't do it for a number of reasons, but the interesting thing is that the first surprising result from the ENCODE project is that most of the genome is transcribed. Spuriously, so very low levels, sort of randomly, and to no real effect because it's not under selection. But most of the genome actually is transcribed. and which means that there is actually a rich toolkit for this kind of thing to happen. It's one of those things where the word junk DNA has such baggage associated with it, because people will point out how these things have come to life. So my point is that, yeah, that's how junk works. Usually it's all crap, but occasionally you'll find something. You ever been to a junkyard and you want a specific part from a car? You can find it and repurpose it and put it in another car. That doesn't mean it wasn't junk. It was by definition junk. It was in the junkyard, and then you find it and you repurpose it. And I think that's actually a very good analogy for how evolution makes use of this stuff. So it's always there, being expressed at a low level. It doesn't matter to you and to me, to any of our cells, but over the fullness of evolutionary time, it adds to the toolkit of something that could be used. Through the accidents of genomic rearrangement, something functional could get created. And that's the beauty of it. That's the nature is endlessly creative, and this is just one example.
A question I have is, you mentioned that there were some human de novo genes that weren't able to study, but couldn't those be inserted into yeast and then study the yeast at that point?
Right. So that's one way to try to find out what a protein does, is to look like who it binds to and who it interacts with. And yeast are a great way to do that. So you can put a bunch of human proteins in the yeast, add one, and then see what comes out and see what sticks to it and that kind of thing. And I'm sure those experiments have been done and are being done. But from a genetic point of view, you really want to manipulate. The problem with that is that you can force interactions that aren't real when you set up an artificial system like that. And also the context of a yeast cell is very different than the context of a human cell. There's a lot, different players. We call that the milieu. The milieu is going to be totally different. So the gold standard in a genetics experiment is to work in the organism and delete the gene or alter the gene, make a temperature sensitive version of the gene, whatever you want, to try to manipulate it. But you just can't do that, because humans are not an experimental organism. But you can take human cells out of the body, of course, grow them in the lab and do that kind of work. But the problem is, with a lot of these things that are in neurons don't grow in the lab very well, right? So you're kind of born with all the neurons you're ever going to have. That's not quite true. You can generate new ones through its stem cells, but a differentiated neuron will never grow or divide again unless it becomes a cancer cell. And so a lot of the experiments that we could do with human cells, we can't do with the kinds of cells that we need to do. So that's why this work goes slow. Humans are the most understood organism in some ways, but not in others, because for ethical reasons, there's a lot of things we just simply can't do with humans.
Great, thank you. Ron Stadler is asking, we were all taught that human and chimp DNA are more than 98% identical. Your findings on human chromosome 21 and other findings from Y chromosome suggest a less than 98%. What is the actual DNA similarity?
I think the 98.8 or 98.7% number is correct in terms of if you just add up the nucleotides like letters in a book, that number holds. But what that number is a very blunt measurement. That's the problem, because it boils everything down to this grand total. You will have some regions of the genome that are even more similar than that are perfectly identical, and then you'll have other regions that are less so. A key part of that number that's missing is that that's only counting base pair substitution. So the kind of mutations that are just simply switching out one base for another, a T for a G or an A for a C or whatever. There are also insertions and deletions, which don't really get captured in the 98% number. It's closer to 99 actually. So that isn't captured indels, and there's more than 4% difference in just the indels. Indels is insertion deletions. We create one word out of that because you can't tell if it was an insertion or deletion. All you know is there's two different numbers. There's more difference in indels than there is in base pair substitutions. And what's interesting is the work that I showed today basically is an indel. So it's in that 4% difference of copy. It's called copy number difference. So we have more copies of a certain stretch of DNA, even though the DNA itself isn't that different. But we have more copies of it. And when you have more copies of things, and this is a lesson we all know well now, when you have more copies of things, that's a recipe for innovation, because when you have extra copies, you can retain the original function with the first copy and then tinker around with the second one without screwing up, whatever was important. So insertions and deletions. So the real story is that the 98% number doesn't really capture all the difference. So the question is very well raised.
Thank you.
Mhm.
Scott Schaffer asks, are any plans to perform the same analysis on chimpanzee chromosomes or those of other apes?
We were only really interested in the human chromosomes at this point. I think that everything we've done would be very easy to do with any of the ape genomes. I don't have any plans to do that myself. The problem, though, is that the other ape genomes are not as well annotated as the human genome. So, the annotation effort is a larger effort than the actual sequencing effort of the human genome. And by annotation, I mean understanding, okay, here's a gene, here's this, here's that, like really mapping it out. And a lot of effort has been put into annotating the human genome. So to do this from the other way around, which is to look at the chimpanzee genome per se, rather than just as a reference, would be a lot tougher, because there are very few chimpanzee geneticists out there. There's some, but very few. But there are literally thousands of scientists working on the human genome and working on getting it annotated. So these things are always harder to look at non-human animals, except for the model organisms like mice and rats and Drosophila. Those genomes we know even better than our own, actually. But chimpanzee is not really a laboratory animal. It is for behavior, but not for genetics. So it would be much harder.
Got it. I won't even try pronouncing this person's name. But the question is, if there's more pointless transcription, would that not mean more risk? And with more risk, more chance to die? Is there not a lot more ways to die than to stay alive?
Okay. So this, as far as I can tell, the spurious transcription doesn't cause any harm. Theoretically, of course, it could, because you could have a transcript that then, runs amok and hurts another protein or another transcript. But that's the beauty of evolution, is the harshness of natural selection is, if that happens, that organism dies and takes it with it. It takes that behavior with them to the grave. So no, spurious transcription doesn't seem to cause any harm. It's sort of just like a background molecular turnover. There's a lot of molecular turnover. I don't know if you know this, but something like 40% of proteins that are built are destroyed almost immediately. 40%. Think of all the calories that we expend on just that wasteful destruction of proteins that we just went to all the trouble to build, and then we destroy them. And one of the reasons we do that is for immune surveillance, so that if there's any viral proteins in there, they get presented to the immune system immediately. That's one reason we do it. Other reasons we do it, remain mysterious. But by and large, spurious transcription translation doesn't cause any trouble. So it's not dangerous in that way.
Makes sense. Scott Schaffer again. Are you able to classify any of what you found in this study as worthy of being included in your next edition of Human Errors?
That's funny. So, I don't know, because this might very well be the opposite of a human error. This might be a case of humans having something kind of special about us that the other apes don't have, that may have even boosted, our cognition and our other features. However, if I had to rewrite, or if there is a new edition of the junk in the genome chapter, one thing I have learned in the process of doing this research is that our genome is an even bigger mess than I thought it was, because I concentrated on the few cool things we found in that mess on the short arm of chromosome 21. The rest of it is really, like I said, just a mess. And luckily, it's a gene desert, so nothing much happened. But if you can imagine, while you're duplicating and this segmental stuff, if you're messing around with important genes, the organisms in which that happened would have suffered because of it, right? So, and they wouldn't be with us anymore. We'd never have detected this. They would have taken that with them again, as I said before. But the fact that segmental duplication is so common, and that this genomic rearrangements have contributed so much to evolution, which means they're not rare. So how many organisms have died? How many human organisms have died over the years because of this sort of crappy control of our own genome? It's impossible to know. But a lot of things that are bad for individuals are good for the species over time, right? Because all of this randomness of tinkering generally hurts the individuals that it tinkers in. But every once in a while, 1 out of every 1,000 has a tinkering event that leads to a new function. And then they become the sort of the founders of that new thing in the species. So evolution is always a tension in what's good for the individual versus what's good long-term for innovation in the species.
Now, do you imagine that miscarriages are often a result of these, problematic changes?
So, we know for sure that the most common cause of first trimester miscarriage, the single most common cause, is chromosomal abnormalities, and having too many chromosomes, too few chromosomes, broken chromosomes. So what I talked about today was segmental duplication, but we also have examples of gross chromosomal changes in our evolutionary past. And most of those, if you can imagine breaking a chromosome and then fixing it in a wrong way, that's almost always going to hurt the organism, right? But every once in a while, it comes up with this new configuration that's beneficial somehow. But the short answer is yes. I think a lot of miscarriages are the result of what we call genetic catastrophe, where there's just a large deleterious change to the chromosomes.
Eugenie Scott has a question. Klein says the difference between anatomically modern humans and predecessors was neurological change with fairly rapid cognitive superiority. Is your work compatible with this, or would you disagree?
I agree 100% with that view. Whether or not my work is a window into that remains to be seen. What we need to do now is to understand what are the real targets of these microRNAs, because no one's done that yet. We have the predicted targets, but we need to find out exactly what the targets are, and there's experimental ways to do that, and we're going to do that when we can. So that's step one. And then step two is to understand how those targets may have changed the evolutionary, the developmental process in the brain. So how the brain takes shape. We know that the sort of big jumps in behavior were not accompanied by big jumps in anatomy. Probably, the wiring, a switch in the wiring wouldn't have left a fossil, really. It's not really brain size that did it as much as the intricacy of the wiring. So whether or not the genes that are the targets of these microRNAs were somehow involved in wiring, I mean, that would be amazing if that were true. So it's possible, and it's to answer the question is yes, it's compatible with that view, and I hope that that we will get to do the work that would kind of give answers to that question. Yeah,
I could see that. Perhaps not for another century, but it could be done.
It takes time. These things do take time, but yeah.
Okay, Teresa Deal asks, what would need to happen for someone with Parkinson's or other fatal disease to volunteer their DNA for study?
Well, that's a great question. So there are many studies going on at any time related to genetics and disease, and you can sign up for these especially if you're in a major city with a medical center like a teaching hospital. They'll have these kinds of studies going on all the time. Genome-wide association studies where they're looking for genetic elements and how they correlate with disease. I've signed up for some of these myself because what they would love to get is someone who has some of these diseases in their family, but they don't have symptoms yet, and they'd like to follow you and see if you do get develop symptoms. So, I mean, I don't have a website I can tell you right now, but if you Google, medical research healthy volunteer, almost all of those teaching hospitals have a clearinghouse, a database of the studies that are ongoing, and you can even choose, like, I'm interested in this disease, or I'm interested in that, and they're looking for volunteers all the time. I've signed up for several myself. In fact, when I was in graduate school, I used to make extra money signing up for medical studies. I know that sounds like a joke, but I totally did it. And because I also knew that they need that, right? They need healthy volunteers to do the work. So I figured I was contributing and making a little money at the same time. And I encourage everybody to volunteer for medical studies. We need healthy volunteers, especially for drug trials and things, because if you think about it, drugs are most urgent in very sick populations who make very poor research subjects because they're ill. So to really understand side effects for a certain drug, you really need a healthy population to do that. So, and I did that. I took, but I didn't take experimental drugs. But they need somebody to take those too. So yeah, you should totally sign up. And I've done the same myself because I have Alzheimer's in my family, and so I've signed up for studies there because if I'm going to go that way, I at least want to contribute to our understanding of how that disease takes shape.
I made my extra money with the psych department for their experiments. Sure. Scott Schaffer again. I received the impression from something you said that embryonic stem cells were unique to humans. Could you speak more to that?
No, definitely not. Embryonic stem cells are all mammals, all the animals have them and use them. What I said was there is a human-specific gene, ESG1, I think is the name of it. I can look it up. That functions in embryonic stem cells in humans, and it has it's very important for keeping their potential. But actually, you raised an interesting question, because I think where you're going with this, or why your brain went that way, is I said this is a human-specific gene. Only humans have it, and it's essential for keeping embryonic stem cells pluripotent, with their full potential. And you're like saying, oh, well, then other creatures, if they don't have that gene, they don't have that. I'm sure they have their own version of that gene. And that's what's kind of cool about this, is that these orphan genes don't represent totally new functions that are unique to an organism. They're just a way that organism does it. And so like with fruit flies, I showed you the experiments that very quickly these new genes become super important to the organism. Because they either replace other genes that were functioning, or they just sort of take on a role. But that doesn't mean that function, that biochemical function, is unique to the organism, right? So embryonic stem cells are found in every animal, and they're crucially important for every animal. Only humans have that particular protein that does the work, and that does the work that way. But other organisms would have their own versions of that protein that does a different version of that job. But good question. I'm glad you caught that, because I didn't mean to imply that.
Mike Vanderman asked, one of our worst characteristics is a lack of empathy for other species. Do you see any hope of correcting that suicidal tendency?
Well, certainly not from this work. But I do wax philosophical a little bit in the final chapter of Human Errors, in the sense that evolution, I mean, I don't have anything really hopeful to say. Evolution has never rewarded a species for discipline and self-restraint and forward thinking and even really for empathy, except within your group. Like, you're designed, we are all very well designed to empathize with those that we consider part of our circle, and you can expand or contract that circle. And I think that's the key to get through the crises that we're in now, is to encourage us to expand our circle and to have empathy across all of our other divisions. But that's not a human problem. Other animals have that, especially social animals identify with a social group. In fact, actually, humans have the most flexible sociality of all. If take this example, so chimpanzees, if you take a chimpanzee from one troop and place them in another troop, someone's going to die, or it's going to be a fight immediately. They have no tolerance for strangers. If they're not familiar with you, you are an enemy in a chimpanzee social group. Humans are the only ones that are tolerant of strangers. Now, we might be suspicious of strangers, but we don't murder them every time we walk into Starbucks. This is the work, by the way, of Mark Moffat, who has a book out, I guess 2 years ago now, called The Human Swarm, who really has very, very insightful observations and data on human sociality. Our empathy is actually the most flexible, and while it is has boundaries to it, our empathy within our group, we also have the ability to expand our group as long as we expose, especially during childhood, we expose children to people from all kinds of different groups. You can build empathy that way. So I'm hopeful in the sense that we have the information that I think we need. I'm pessimistic in the sense that we seem very resistant to use that information. The same thing, we have all the information we need on renewable energy and sustainable. We have all that. We're not waiting on the science for any of these things, right? We have that. We're lacking the will. So we'll see.
Yeah, I definitely agree that the expansion of our circle of empathy is so important.
So important. So important.
Is that the last of the questions? It seems to be. So thank you again for your presentation. I loved it. I'm sure the rest of the group did as well. So for the rest of you, do not forget to mark your calendar March 11th, Thursday evening, for our next Skep Talk, which will be Daniel Loxton. He's a Canadian writer. He's an illustrator and a skeptics. And thank you all for joining us. This has been very, very good. Stay well.